my 6th video in my AP Biology review series, and it is about cellular respiration and photosynthesis. I'm going to start with redox reactions because these are important to both processes. Redox reactions are oxidation and reduction reactions. Oxidation is the loss of electrons from one substance. The substance that loses electrons becomes oxidized and is also called the reducing agent because it reduces something else. Reduction is the opposite. It's the addition of electrons to a substance, and it's called reduced because it becomes less positive. The substance that gains the electron becomes reduced and is also the oxidizing agent. I hope you can see that oxidation and reduction will always happen together because one substance will donate an electron and another one will accept it. Here's a simple example of that. We have the sodium atom and a chlorine atom. The sodium atom will become oxidized because it gave an electron away to chlorine, and chlorine will become reduced because it accepted an electron and is now less positive. Cellular respiration occurs in all cells, including eukaryotes and prokaryotes. Here is the overview equation for it. We have sugar and oxygen yielding carbon dioxide, water, and energy. And here is that equation using the formulas for all the compounds. I like this picture because you can see how all the atoms will be rearranged from sugar and oxygen in order to yield energy. It's a catabolic pathway because molecules are being broken down, and it's exergonic because energy is released. The delta G, or change in free energy, is going to be negative because we start with more energy than we end up with, and energy is released. To be specific, it's around negative 686 kilocalories per mole. Glycolysis occurs whether oxygen is present or not, and it occurs in the cytosol. Glucose, which is a 6-carbon compound, is split and its atoms are rearranged to form 2 molecules of pyruvate, which is a 3-carbon compound. There's a net production of 2 ATP, and I say net production because we have 4 produced really, but 2 of them will be used in the process. And these ATP molecules will be produced by substrate level phosphorylation, and here's a picture of that. We have a substrate with the phosphate group and an enzyme which will catalyze the reaction so that the phosphate group gets transferred to ADP to form an ATP and the product. There's also a net production of 2 NADH molecules because in this process, NAD+ will be reduced, right? It's going to gain an electron and become NADH. And I circled them because they're important to continuing glycolysis. We'll talk about that more in a few slides. Again, glycolysis does not require oxygen to occur, which is why it's anaerobic. If there's oxygen present, the cell will undergo aerobic respiration, which is that top pathway, and go on to the Krebs cycle, and then the electron transport chain and chemiosmosis. If there's no oxygen present, it'll undergo anaerobic respiration and go down that lower path to fermentation. As you can see, a lot more ATP will be produced during aerobic respiration, so obviously if oxygen is present, that would happen, but if there's no oxygen, fermentation will have to occur. Anaerobic respiration, again, occurs when oxygen is not present, and it occurs in the cytoplasm of the cell. No ATP will be created. The goal, the reason it happens, is to regenerate NAD+ in order to continue glycolysis. So that's why I had circled the NAD+ molecules in the glycolysis picture, because that's really what helps that process continue on. And even though glycolysis doesn't create a lot of ATP molecules, it does generate 2 per glucose. So that is an important source of energy for the cell. For 2 pyruvate molecules, so 1 glucose, 2 NADHs will be made to NAD pluses through fermentation. And again, this would occur after glycolysis. There's lactic acid fermentation, which changes pyruvate into lactate. This occurs in humans. You've felt this before. For example, let's say you've been doing a lot of heavy exercise and then your muscles start to hurt. That's because you're not letting your muscle cells have enough oxygen to undergo aerobic respiration, so they start undergoing fermentation and producing that lactic acid, which is why you feel that pain. It's the acid that's hurting you. And in the picture you can see it's just an overview, but the important thing to keep in mind is that regeneration of NAD+ to be used in glycolysis, to continue the process of glycolysis so that we continue to have ATP for the cell to use. There's also alcoholic fermentation, which gives off carbon dioxide. There's 2 steps to it, and it results in ethanol. So here we have carbon dioxide being removed, an intermediate molecule, and then ethanol. The Krebs, or citric acid cycle, same thing, occurs in the matrix, which is the inner space of the mitochondria. Pyruvate is converted into acetyl coenzyme A, and this occurs before it gets to the citric acid cycle. So again, pyruvate will be made in the cytoplasm, and as it's being transported into the mitochondria, it will be changed into acetyl coenzyme A. Acetyl coenzyme A is the first molecule to enter the cycle. The molecule will be converted to many intermediate molecules and oxidized throughout. And this oxidation will lead to the reduction of 3 NAD+ molecules to NADHs and 1 FAD to FADH2. And these molecules are important because they will provide electrons during the electron transport chain, which is the next step of aerobic respiration. Carbon dioxide will be released. 1 ATP molecule will be formed through substrate level phosphorylation, and all these numbers are per pyruvate molecule. But we should keep in mind that the cycle will turn 2 times per glucose molecule, so once per pyruvate. Next, we have the electron transport chain. This happens in the inner membrane of the mitochondria. Keep in mind that mitochondria has 2 membranes. The NADH and FADH2, remember that we created in the citric acid cycle, will donate electrons to cytochromes. And cytochromes are proteins that are in the inner membrane. They have a heme group that accepts and donates electrons. The cytochromes will pump hydrogen ions from the matrix into the inner membrane space against their concentration gradient. The fact that it's against the concentration gradient will be very important, and we'll talk about that in the next slide. Oxygen is reduced and will bond to hydrogen ions to create water as a product. Chemiosmosis. Hydrogen ions will diffuse down their concentration gradient through ATP synthase. So remember, we said in the last slide, they were pumped against their concentration gradient, so now they want to diffuse back. And they will diffuse through ATP synthase. It's important to note that the internal membrane of mitochondria is impermeable to hydrogen ions, so they can't just simply diffuse through it. They need to use a protein. And in this case, they'll use ATP synthase, which is a complex protein in the membrane. The diffusion of hydrogen ions will power ATP synthesis. And this is called oxidative phosphorylation. Remember in the past we were saying substrate level phosphorylation, that's one type, but this is oxidative phosphorylation because it's powered by redox reactions. And around 32 or 34 ATP molecules will be produced, and this is per glucose in the electron transport chain in chemiosmosis. Next, we have photosynthesis, which is performed by photoautotrophs. They're called photoautotrophs because they use light, so photo, and they're autotrophs because they make their own food. They're producers. Photosynthesis occurs in the chloroplast. There's a picture of one. And this is the formula for it. We have carbon dioxide and water plus light energy yielding sugar, water, and oxygen. So keep in mind, like, yes, there's water on both sides, but that's because water is needed for it to happen, but it also does produce water. It has two stages, the light reactions, so that's the photo part of photosynthesis, and the Calvin cycle, which is the synthesis part of photosynthesis. Here's a picture of stomata. It's important to keep in mind that plants will exchange gases with their environment through their stomata, so that's where oxygen will exit and where carbon dioxide will enter. Pigments. Pigments absorb light energy. So chlorophyll A and B are both green, but there's also other accessory pigments that absorb colors such as yellow, orange, and red. The accessory pigments expand the spectrum of light that can be absorbed to power photosynthesis. Absorption spectrum is the graph that shows light absorbance versus wavelength. Here's an example for chlorophyll A and B. As you can see, the graph showing the absorption is lowest when the wavelength is in that green area. And that means that it's not absorbing a lot of green, which means it's reflecting a lot of green. So that's why it appears green, because it's not absorbing that. The light reactions occur in the thylakoid membrane of the chloroplast. A photon of light strikes and is absorbed by a chlorophyll molecule in photosystem 2, and it excites an electron to go to a higher energy level. Water is split and replaces the electron that photosystem 2 lost when it went to a higher energy level. And the electrons from photosystem 2 will go through an electron transport chain, that's what ETC stands for, to photosystem 1. The electron transport chain is made of cytochromes. And keep in mind that hydrogen ions are getting pumped into the lumen, which has a high concentration of hydrogen ions. So they're getting pumped against the concentration gradient, which might remind you of what we learned in cellular respiration. NAD plus is reduced to NADPH. Hydrogen ions will diffuse through ATP synthase into the stroma, and that diffusion will power ATP synthesis. NADPH and ATP are important because they'll be needed in the Calvin cycle. And what we talked about was non-cyclic photophosphorylation, because it's not a cycle, it's one thing to the next. However, there is something called cyclic photophosphorylation, which only replenishes ATP, and this will be used when the Calvin cycle, which uses a lot of ATP, uses a lot of energy. When it uses all of its available ATP, it'll undergo cyclic photophosphorylation to regenerate ATP needed. The Calvin cycle occurs in the stroma. The goal is to make sugar using energy from ATP and NADPH, so that's why we call it like synthesis, part of photosynthesis, because here is where the sugar is made. It can be broken apart into 3 phases. Carbon fixation, which is where the carbon dioxide molecule enter the cycle and will be bonded to ribulose biphosphate. Rubisco is the enzyme that catalyzes this, and Rubisco is an important and I don't want to say popular, but it's found a lot, a very common enzyme. Phase 2 is reduction. NADPH will reduce the molecule, and the output is G3P, which is a sugar. You might be thinking, wait, doesn't it make glucose? Well, yes, but not directly. the direct output is G3P, which is glyceraldehyde 3-phosphate, which will then become glucose later. Phase 3 is the regeneration of the carbon dioxide acceptor. So ribulose biphosphate is remade. Remember, that's what was in phase 1. That's what carbon dioxide will bind to. And a while ago, some people used to call the Calvin cycle the dark reactions. But that's not really accurate because this also occurs in the light. Keep in mind that it does need the products from the light reactions. So this does not occur in the dark. Photorespiration. So let's start with C3 plants. That's what we were talking about. It's the majority of plants. And this is where carbon will be fixed by rubisco, that enzyme I mentioned. And it's called a C3 plant because the first product is a 3-carbon molecule. But keep in mind that stomata will be partially closed on hot days. And that means that the amount of sugar produced will decline because the cell will be deprived of carbon dioxide, because stomata is where carbon dioxide will be able to enter the plant from. So what happens is that rubisco will bind to oxygen instead of carbon dioxide. And that doesn't allow the Calvin cycle to happen like it should. And that's called photorespiration. And after rubisco binds to oxygen, it creates a molecule that will be split, and then carbon dioxide will be released. So that's why it's called photorespiration, because photo, it occurs in the light, and respiration, oxygen will be consumed while carbon dioxide is given off as a product. But this process only consumes ATP, it doesn't help the plant in any way. So you might be wondering why? Why does this exist? Why is there a process that's so inefficient that evolution has not gotten rid of? Well, some think it's because the ancient atmosphere had lower levels of oxygen and higher levels of carbon dioxide, so this wasn't a problem. Rubisco being able to bind to oxygen was not a problem. But of course, there's a solution to this problem, basically. C4 plants have a different plant anatomy. Examples include sugarcane and corn. They have a different process of carbon fixation that happens before the Calvin cycle. The first product is a 4-carbon molecule, which is why it's called a C4 plant. The enzyme PEP carboxylase adds to carbon dioxide PEP in mesophyll cells. PEP is phosphenylpyruvate. And what's important about PEP carboxylase is that it has a much higher affinity for carbon dioxide than for oxygen. So it won't happen like it did with rubisco. Rubisco likes to bind to oxygen as well, but this PEP carboxylase won't bind to oxygen, which is important. So the mesophyll cell is where this happens. Here's a picture of the plant anatomy of a C4 plant. You can see that first, this side A of the box at the bottom will occur in the mesophyll cell. Then the second part will happen in the bundle sheath cell. Carbon dioxide will be pumped into the bundle sheath cells. And this is important because it makes sure that carbon dioxide is the molecule that will enter the Calvin cycle. It's not possible for oxygen to do so. And again, the goal is to reduce photorespiration, and this is especially important in regions that are hot and dry. Cam plants are another type of plant that have basically adapted to their environment. These are plants that live in the desert. For example, cacti and pineapples. Their stomata are closed during the day and open at night. That's why I have that graph there at the top. That graph shows the time when that plant absorbs the most carbon dioxide, and it absorbs the most carbon dioxide during the night to be specific, between around 4 a.m. and 8 a.m. That's when it absorbs it. So that's when the stomata are open. And they incorporate carbon dioxide into organic acids during the night. That's why it's called cryolysine acid metabolism. The acid part comes from the fact that it incorporates carbon dioxide into those organic acids, and that's what CAM stands for. The mesophyll cells store organic acids until the day, and during the day, carbon dioxide is released from the organic acids and used in the Calvin cycle. And keep in mind, during the day, the light reactions will still occur. They'll supply the ATP and NADPH needed. So these plants are unique because their stomata are closed during the day. But again, this is all to improve efficiency. Thank you so much for watching, and please subscribe if you would like to see more videos.